In this video, we are going to use some of the underlying theories of DNA replication to figure out the base sequence. Okay, so 7.111 says outline the use of nucleotides containing, what is this? Dideoxyribonucleic acid. So all that's saying is it's basically DNA, right? Deoxyribonucleic acid is DNA, but it has two deoxys. And what that means is instead of having one H here, we've added an H over here. So there's two H's connected to carbon 1, carbon 2, and there's two H's connected to carbon number 3. And we're going to talk about why that's important in a second. Well, I guess I'll mention it right now. So what it does is it actually stops DNA replication. So that by allowing it to stop DNA replication, we're able to figure out the base sequence. And I'm going to demonstrate this on the next slide. But let's talk about this molecule first. So this is DNA. So remember the nucleotide has carbon 1, 2, 3, 4, carbon 5 connected to our phosphate, which we normally just draw as a circle. And over here, they have adenine here, but we normally draw it as a rectangle. So there's our molecule of DNA that we're comfortable with. Remember that DNA, this is carbon number 5, right? DNA replicates from the 5' to the 3', here's carbon number 3, in the 3' direction. And that simply means that the next nucleotide would be added here. That's all that means. Carbon 5, carbon 3, and we just keep adding nucleotides to the bottom, right? If you want to think of it as the bottom, to the bottom of the nucleotide above it. And it can do it because it forms, right? It forms a covalent bond through a condensation reaction with this OH group. But if there's no OH group, the next nucleotide that tries to come, and this example, dideoxy, and a lot of times we just, we call it like DNA. So we put an extra D in front of it. The next nucleotide can't connect to it because it can't form that condensation reaction because we don't have that OH. So that's how it stops the DNA replication. And on the next slide, I'm going to show how we can figure out the base sequence. So that means which letters first, which letters second, maybe another A is third. So what is the sequence in our DNA? All right, so let's try this. So in this diagram, it might be a little bit confusing, but hopefully we can figure this out. So this base sequence here, I want you to pretend that it's a mystery. We don't know what it is, okay? And we want to determine what is the sequence of this strand of DNA. We know that DNA is going to replicate in the 5' to 3' direction. So our first base will attach there, our second base will attach there, third, fourth, fifth, and that's how we will replicate our DNA. Now, we can use this dideoxyribonucleic acid to determine the sequence. So what they do to each one of them, depending on the base attached to it, they'll put a different colored marker on it. So let's pretend that our first dideoxyribonucleic acid is guanine. All right. So guanine comes on here. Oh, look at that. Guanine is going to form a hydrogen bond with its buddy, cytosine. Now, because guanine is the dideoxyribonucleic acid, we cannot make the chain any longer. So what they do is they look at this piece of DNA and they run it through something called a gel electrophoresis. This is a machine we'll talk about later, but this is a machine that tells you how long the pieces are. And shorter pieces or smaller pieces of DNA end up making it further. DNA is slightly negatively charged, so it's attracted to this positive side, and they travel farther and faster. So we can look here that our smallest piece of DNA is this blue one, so that must mean the first base is guanine. Now, I don't really like this diagram because it looks like the next one we try is A, but they normally don't do it that way. Then they do it again, okay? It's really confusing about how they allow it to happen again. It has to do with concentrations and whatever, but let's not worry about that. So we run the machine again. A will form a bond here, no problem, because it's just a normal nucleotide, because the only dideoxys we've added to the mixture is guanine. So A will form a bond here, and then again, the next one is cytosine, and then it's thymine, right? And then here, we will have another guanine. And this guanine is like somewhere medium-sized, right? So you can see that it stopped somewhere here. So if we're looking at this diagram, 1, 2, 3, 4, 5, it should be 1, 2, 3, 4. The fifth base should be guanine. And we do it again. Adenine, adenine, guanine again. So here we have guanine, and that's a pretty long piece. The third longest, right? The longest, second longest, the third longest piece of DNA. And then we know the positions of where guanine is on this segment of DNA. Let's do it again. This time, let's do adenine. Adenine is green. We'll put a green colored marker on it. So I'll change the color of my crayon. So there's adenine. The first pair, here we form a hydrogen bond with thymine. And look at that. That's our second shortest, only 2 bases long. Our second shortest is adenine. And then we do it again. Guanine, adenine, cytosine, thymine, guanine, adenine. So we have an adenine here, about medium size. And then when we run it again, we have another adenine here, which was represented here. And then you repeat the whole thing over again. Let's do cytosine. Cytosine, we'll make orange, I think that color is. That looks like it's yellow, but who cares? And then cytosine here, the third shortest. Look at that. Cytosine is the third shortest thing that showed up in our sample. Thymine, guanine, adenine, guanine. Oh, another cytosine here, almost the longest one. Here we have cytosine. And then the last one, which will be thymine. We'll make it red. And then it goes along. It stops because another one can't connect to it. We run the machine again. We run the Sanger model again, and we get another one down here. So we have a thymine that's 1, 2, 3, 4 bases long. 1, 2, 3, 4 bases long. And then this one all the way down here is the longest one. So that is how they determine the base sequence. And you can just look at this diagram here, and we could say, the first one is guanine. The second longest is shortest. The second shortest is adenine. The third shortest is cytosine. The fourth shortest is thymine, and so on. So we can get the sequence of DNA from shortest to longest. And from this, once we complementary base pair them, because guanine always attaches to cytosine, then we can determine the original sequence of CTGACTTCGA, CTGACTTCGA. And that is how you can figure out the base pair sequence by having DNA replication stop through the use of deoxyribonucleic acid. I'm sorry, dideoxy. A little confusing, maybe. There's some good questions with this that I'll practice you with. I will practice with you. And if you can get those, you totally nailed it.